Hello everyone, this is Professor Manohar. Today we are going to discuss about the carbohydrates. So first of all, let us see how much we know about the carbohydrates. Okay, so let me ask you some questions. Can diabetic patients drink cool drinks? Yes or no? How fruit intake decreases the constipation? Why humans cannot digest cellulose? Why dextrose is injected to patients? Difference between DNA and RNA? How cosmetic surgery is done? And how to check your kidney functioning. Okay, so by understanding this topic, carbohydrates, you are going to get answers for each and every question. Okay, so a small request that you don't consider marks while you are learning the topic. Okay, so today we are going to cover the introduction, classification like monosaccharides, disaccharides, oligosaccharides and polysaccharides, and its arrangement and all. Then we are going to discuss about the biological importance of the carbohydrates. So where is going to be located and what is function and what is the benefit we are going to get. Okay, and last we are going to discuss about the identification test for the carbohydrates. Okay, so jump to these carbohydrates. So this is polyhydroxyaldehyde and ketones normally. So here depending on the monomers, so we can categorize the carbohydrates into monosaccharides, oligosaccharides, polysaccharides, etc. So if you see this monosaccharides based on the functional group present on this monosaccharides, we can categorize as aldoses. If aldehyde group is there, normally we'll categorize as aldoses, example glucose. If the ketone group is there, so normally we'll categorize as ketoses, and example is fructose. Apart from the functional group, we can divide the monosaccharides based on the carbons present on this. So if total 3 carbons are there, normally we'll call it as trioses. If 4 carbons are there, we'll call it as tetroses. If 5 pentoses, 6 hexoses and 7 heptoses. Here depending on the number of the carbon present on the monomer, we can categorize the monosaccharides into trioses, tetroses, pentoses, hexoses and heptoses. Yeah, next. So here if the two monomers are bind together, we'll call it as disaccharides. So example for the disaccharides are maltose, lactose and sucrose. Next, trisaccharides. So here 3 monomers are going to be joined together is raffinose, tetrasaccharides, stachyose and polysaccharides. Here the number of the monomers if joined more than 10, it is said to be the polysaccharides. So if the same type of monomer is joined together, it is called as homopolysaccharide. So example starch, dextrin, glycogen, cellulose, inulin. So we are going to see the structures of these and we are going to explain the biological importance of these agents. Next, heteropolysaccharides. If the monomers are of different categories, so in the sense glucose fructose or glucose galactose, so these are going to be a chain more than 10 monomers. It is called as heteropolysaccharides. Example hyaluronic acid, heparin, all these agents, chondroitin, dermatan and keratin, all these are the example for the polysaccharides. So here just you need to understand that depending on the number of glucose moiety, so we can categorize as monosaccharides, oligosaccharides and polysaccharides. Then so we'll see the monosaccharides. So monosaccharides are examples are the glucose, fructose, galactose, etc. Okay, so here during the digestion the starch is partially transformed into the maltose by the pancreatic enzymes. Then maltose is going to be further digested to the and yields two glucose molecules. So here the ultimate aim of the digestion is to make the monomers so that it can easily absorbable. Okay, so here you can see the pictures of the dextrose. Here some of the commercial products. Here when the patient is ill, when he is not having any energy, so normally we'll give this dextrose through IV route. So the basic purpose is, so we are going to give the readily energy to the system. Okay, so next Tata glucon, so which is consisting of the glucose and water, and glucon D, which is a purely form of the glucose. Okay, here you need to understand that the only monomer present in is glucose. Okay, so here sucrose, all these are sweet in taste, right? Glucose or sucrose, fructose, all are sweet in taste. So here I want to discuss about this artificial sweetener, which is not at all a carbohydrate. Okay, but still you need to understand that here the saccharine, it is named because of the sweetness of this. Okay, so this is not belongs to carbohydrates at all, and it will not provide any energy, no energy, you can see here. Then but the taste is going to be 300 to 400 times as sweeter than the sucrose. Okay, so but it has the bitter and metallic taste at high concentration. Okay, so normally what they will do is they will mix these different sugars along with the saccharine, and they will prepare various goods to mask this bitterness and metallic taste. Okay, saccharine is a chemically inert and heat stable chemical. It is an acid form, and saccharine is water insoluble. It is water insoluble, but the salts of saccharine like sodium and calcium, so it will enhance the solubility. Okay, so here I mean to say, so when you are using saccharine, so wantedly or unwantedly you are adding the calcium and sodium, so which is not at all good for the individual who is suffering with blood pressure or the kidney stones, etc. Next, saccharine is used to sweeten the products such as drinks, candies, cookies and medicines. Okay, here the basic question is the diabetic patients can take the cool drinks? So if these cool drinks consisting of the high quantity of the saccharine, obviously yes, because it doesn't have any carbohydrates, no sugars are there, and it has no food energy even. But the problem is here various companies are going to mix these various sugars to mask this metallic taste. They are going to mix with saccharine with some natural sugar, so that may create some problem. But most of this all these cool drinks or the artificial sugars are used in preparation of various candies like and cookies, etc. Okay, so ultimately answer is yes, diabetic patients can drink the cool drinks if the saccharine used as a sweetener. Okay, so here now we'll come to our original glucose moiety. Okay, it is the most abundant monosaccharide. It is present in two forms, dextro form and levo form, depending on its arrangement. Okay, so here you can see, so here OH group is in dextro form, right side, so normally we'll call it as D-glucose. If the OH groups are in left side, so we'll call it as levo form. And moreover, so these linear form can rearranged in a circular form. Okay, it can rearrange, and it can form the alpha and beta glucose. So the basic logic of alpha and beta glucose is, if it is formed in a cyclic form, so it will appear like a plane. Okay, so if hydrogen group is above this plane, it is said to be the alpha, above alpha, and if the hydrogen group is below, normally we will call it as beta group. Okay, beta glucose. Okay, so here alpha glucose and beta glucose is going to be depend on the hydrogen group present at the carbon number 1. So this is carbon number 1. So at carbon number 1, if hydrogen is present above the plane, it is called as alpha glucose. If the hydrogen is at the 1, if present below the plane, it is said to be the beta glucose. Okay, so here I plan to explain like how this cyclation is going to be take place. So here if the moiety is tilt a little bit, so it's going to be appear somewhat like this. So here this OH group is going to be donate its hydrogen to the carbon number 1. So resulting of that, there will be a bond is going to be formed, which is a glucose moiety cyclic monosaccharide. We can call it as cyclic monosaccharides, 6-membered. So in case of the fructose, it is 5-membered will be there. Okay, so here how the glucose is converted to the linear to cyclic form. So here, so because of the internal rearrangement, okay, here there is no addition or deletion of any atoms, only the internal rearrangement will makes the cyclic 6-numbered monosaccharides. Clear? Then we'll move to the fructose. Fructose, it is occurs naturally in other plants foods such as honey, sugar beet, sugar cane, vegetables, etc. Fructose is the sweetest naturally occurring carbohydrate. It is 1.2 to 1.8 times sweeter than the sucrose. Okay, so sucrose ultimately is going to be a combination of the glucose and fructose. If the fructose only taken, obviously the sweetness is going to be very high. So even in fructose, we can see the dextro and levo forms. If it is right side OH group, normally we will call it as dextro. Then if it is left, it is called as levo. Okay, so here after cyclization, so here there will be a formation of the pentameric cyclic. Okay, so pentameric cyclic is going to be formed. So here, but you can count the number of the carbons are going to be 6. So here 1, 2, say here will from here will come 1, 2, 3, 4, 5 and 6. Okay, fructose consisting of 6 carbons but pentameric in structure. That you need to understand. Next, galactose. So here galactose is a monosaccharide and has a same chemical formula as glucose. So galactose is a major dietary sugar for humans. The hydrolysis of the disaccharides lactose yield galactose and glucose. So here you can see the structure of the glucose and galactose, which is completely similar except at carbon number 4. So here the plane is going to be changed. So a hydroxyl group is going to be present upon the plane. It is said to be galactose, or if OH group is below the plane, it is called as glucose. So apart from that, everything is the same. Okay, so here the glucose and galactose, the difference is only at position 4. The orientation of this H and OH group are going to be changed. Clear? Next we'll come for the one more important sugar, which is monomer. Okay, so this is a pentose sugar which has 5 carbon atoms and 10 hydrogen atoms. It is a molecular formula. Okay, so C5H10O5. This is also known as aldopentoses, as it has the aldehyde group attached to the end of the chain. Okay, so here you can see the 5 carbons. So here if it is ribose sugar, so this ribose sugar is going to be present in the RNA structure. This is a backbone, so which is bind with this phosphate group as well as the nitrogen bases. Okay, so RNA, so main difference between the RNA and DNA is at position 2, so OH group, so at carbon 2, okay, obviously, so there won't be any OH group, so this is called as deoxyribose, which is present in the DNA. So ribose is going to be present in the RNA. Okay, so here one of our question is, so what is the basic, there are some other differences are there in DNA and RNA, but in case of the ribose sugars, so normal ribose sugar is going to be present in this RNA, and deoxyribose is going to be present in the DNA. Clear? Next one of the fascinating and important one, the disaccharides. It's composed of two molecules of the simple sugar linked with the glycosidic bond. Disaccharides found in food including sucrose, lactose, maltose, grain sugar, all these consisting of the glucose. Okay, so here we'll see, like we'd like to see like what are the different disaccharides present and its structure. So normally the maltose, lactose and sucrose, these are the three disaccharides present in the. So here we'll discuss about the maltose. Maltose is a simple glucose. Two moieties are going to be bind with the glycosidic bondage. Okay, so here you can see this is one molecule of the glucose and this is second molecule of the glucose, which is binded with this glycosidic bond. Okay, so we'll explain further like what is glycosidic bond. So here if the hydrogen is there up, it is called as alpha, right? So this is alpha glycosidic bond, alpha, and this is carbon number 1. So 1 to 4 alpha glycosidic bond is going to be present in the maltose. Next, coming to this lactose. So lactose hydrogen group is below the plane, so normally it is called as beta. Okay, beta galactose and the alpha glucose. So the combination of this beta galactose and alpha glucose, so that is said to be the lactose. Next, sucrose. Sucrose is a combination of glucose and fructose. Okay, again by the glycosidic bondage. So let's see like how this glycoside bond is going to be formed. So here normally the condensation reactions where the two molecules are going to be combined together by releasing of the water molecule. First we'll see what happened in case of the lactose. So lactose, so here we can see the structures of the glucose and galactose. Only difference is the arrangement of this OH and H group. Okay, so here there will be removing of the water molecule between this. So here this is the beta galactose and alpha glucose. So here because of the removing of this particular water molecule, the lactose is going to be formed. So here the lactose beta glycosidic linkage, this particular linkage is called as beta glycosidic. Next, formation of the glycosidic bond between the glucose and fructose. So here these consisting of the same is going to be present in the same plane and the water molecule is removed. So because of that there is a bond between alpha and beta. So normally alpha D glucopyranol and beta D fructofuranide 5-membered ring. So this bond is going to be formed between the sucrose. So coming to this glycosidic bond between the glucose and glucose in case of the maltose. So here the maltose is going to be present is both are alpha glucose moieties, and again by the removing or condensation reaction, so removing of the water moiety, it can form the maltase. The bond is alpha 1,4 glycosidic bond. Okay. So next, osozone formation. Ozozones are a class of carbohydrate derivatives found when reducing sugars are reacted with excess of the phenylhydrazine at boiling temperature. Okay, so here when these ozozones are going to be needle shaped crystals, so you can see in glucose, fructose and even in mannose. So all these are needle like structures can be seen. So ball like structures we can see in case of the galactose. Then free long needle shaped crystals are going to be seen in xylose, and sunflower shape is going to be seen in maltose. So depending on this ozone formation, we can identify the type of the monosaccharide. Okay, next we'll go for the oligosaccharides. Oligosaccharides, which are these monomers are more than 2. It may be 3, 4, 5, 6, depends. Okay, so oligosaccharides. So what is the basic use of this oligosaccharide? So these are the carbohydrates of from 3 to 6 units of the simple sugars. Example, raffinose and stachyose. So raffinose consisting of 3 monomers and stachyose consisting of 4 monomers. So these trisaccharides are normally galactose, glucose, fructose in case of the raffinose. Raffinose, so consisting of if it is hydrolyzed, it may yields galactose, glucose and fructose. Okay, next we'll see the uses. The fructo oligosaccharides are commonly used for the constipation. So here to relieve the constipation, so we'll use this. So that is the reason why the clinician will advise us to take lot of vegetables and fruits, because these vegetables and fruits consisting of these oligosaccharides which cannot be further digested, but it will increase the water content in your fecal matter. So resulting of that, the smooth passage of the fecal can be take place. So that is nothing but the treatment of the constipation. So since these are not going to be absorbed by our system, so here the individual cannot gain weight, and it will also use to prevent the Travel's disease, diarrhea, and to treat high cholesterol level and osteoporosis. Oligosaccharides such as fructo-oligosaccharides and galacto-oligosaccharides are known as prebiotics. They serve as a nutrition for the trillions of the microbes residing in your gut. So here this is a good food for the microbes which are there in your stomach, microbial flora. So it can grow enormously and it will produces it will helpful for a proper digestion and absorption of the food. And these microbial flora is helpful to secrete various vitamins, B complex vitamins, so which can be absorbed by our gut. So I mean to say these are the very important substance one must take. So here we'll see like what are the beautiful fruits and vegetables in which the oligosaccharides are going to be present. So if you see this example, blueberries, pear, watermelon, nectarine. Coming to vegetables, garlic, spiring garlic, leek, white onions, and all these vegetables and fruits consisting of these oligosaccharides, which consisting of lot of oligosaccharides, so that helps a proper digestion of the food, at the same time to relieve the discomfort because of the constipation. Next we'll move to the polysaccharides. Polysaccharides are the polymers of the monosaccharides. So it is made up of glucose unity, glucose or fructose monomers. The polysaccharides basic function are the energy storage and structural support. The starch and glycogen are highly compact polymers that are used for the energy storage. The cellulose and chitin are a linear polymer that are used for the structural support in plants and animals respectively. So we'll see like what is the basic use of these polysaccharides. So first one is glycogen. Glycogen, it is used as a carbohydrate stored in animal. So normally glycogen is going to be stored in the liver. So it is a polymer of alpha glucose 1 with alpha 1,4 glycosidic bond and alpha 1,6 glycosidic linkage. So here I mean to say, so here alpha 1,2,4 glycosidic linkage, and in branching it will be alpha 1,2,6 glycosidic bond is going to be available. So the main difference between the starch and the glycogen is the with glycogen within 6 to 8 monomers there will be a branching is going to be take place. I mean to say number of branches are going to be high in case of the glycogen, whereas in starch here the branching is going to be it will branching is going to take place at 20 monomers. After 20 monomers, here the branching density is going to be less in case of the starch. The basic chemical formula is C6H10O5, and starch is a polysaccharide composed of the glucose monomers joined in alpha 1,4 linkage. The simplest form of the starch is the linear polymer amylose and amylopectin. So this is the structure of amylose, which is a linear form, and amylopectin, so you can see a lot of branching. So all this together combined so will form the starch. So here you can understand alpha 1,2 glycosidic bond and alpha 1,6 glycosidic bond is going to be present as like the glycogen. So here coming to the structure wise, there is no difference between the glycogen and starch except the branching density. So starch is the main energy storing in the plants. Okay, so it only like in the starch in the plants, whereas glycogen only in the animals. So that is the basic difference between the starch and glycogen. So here these are the various sources of the starch: rice, wheat bread, corn, potato. All these are the starch like source of the starch. Okay, so next we'll go for like why cellulose is not digested by human but animals can. Okay, so if you see this cow, buffalo, cow, goat, sheep, all can digest the grass, but human cannot digest the grass in the sense cellulose. So let's see like why. So cellulose is the most important structural polysaccharide present in plant. It is made up of unbranched chains of the glucose molecules linked via beta 1,4 glycosidic bond. Every alternate glucose molecule in cellulose chain is inverted. These chains are arranged parallel to each other to form the microfibrils. So here the main character is normally in glycogen or starch we saw alpha 1,2,4 glycosidic bond, where in cellulose we here the bond is beta 1,2,4 glycosidic bond. So this is the reason why the human cannot digest the cellulose. Okay, so and moreover, if you see the structure, so these are going to be arranged in a tightly fashion with the interhydrogen bonds. The glucose chains lay side by side to make a thick and strong structure. So here you can see the if you magnify this particular thing, so you can see the hydrogen bonds are going to be present between different strands. Okay, so on the whole, if you see the difference in structure and arrangement of the glucose in different carbohydrates, so in starch amylose and amylopectin is going to be there. In glycogen the number of the branching is going to be there, though it is made up of same glucose, so branches are going to be there. So here the cellulose also consisting of the same glucose, but only beta bonding. Here alpha bonding is going to be there, but in cellulose it is the beta bonding is going to be there. So this is the basic difference between the starch, glycogen and cellulose. Oh, so what is this guy doing? So he's after his heavy work, he's applying grease to this instrument so that the friction can be reduced and it can work properly. So this is work similarly somewhat like our hand, right? Even appearance also it will look like our hand. So for this instrument we are applying this grease every day after every work. So what about our hand? So here grease is not required for our hand. Yes, there is a natural grease is going to be there in our joints. That is nothing but hyaluronic acid, which is a polymer of glucose. Okay, it is an anionic non-sulfated glycosamine glycate distributed widely throughout the connective epithelial and neural tissue. Hyaluronic acid is a major component of the synovial fluid and was found to increase the viscosity of the fluid. So it will act as a shock absorbent. Okay, hyaluronic acid is a polymer of disaccharides which are composed of the D-gluconic acid and N-acetyl glucosamine linked via the alternate beta 124 and beta 123 glycosidic bonds. So because of this structure, a thick viscous fluid is going to be present in our joints that may act as a grease, okay, which will reduce the friction when you are moving your joints. So this is the beauty of the nature. Next we'll see so due to its high biocompatibility and its common presence in the extracellular matrix of the tissue, hyaluronase is gaining popularity as a biomaterial scaffold in tissue engineering research. So here these are the naturally available polymer, so it will not do any harm. So and moreover it has the ability to absorb the water molecule and to bulge. So normally to elevate this it will act as it will absorb the moisture. So when you injected these at different areas, so it will swell and it will create some tightening effect. So here you can see the wrinkles in this particular picture. So after treating, before treating, after 4 weeks of treating and after 8 weeks of the treatment, all the wrinkles are almost gone. So coming to the Anushka Sharma, so here this is the normal lips before the application of this hyaluronic acid, and this is after application of the hyaluronic acid. So you can see some elevation in the lips. The same thing can be seen in this picture also. Okay, so next polysaccharide is dextrane. Dextrane normally it's a plasma expander. Okay, it's a high molecular weight. Dextrane is a plasma volume expander made from the natural source of the sugar. It works by the restoring the blood pressure lost through the severe bleeding. The severe blood loss can decrease the oxygen level and can lead to the organ failure, brain damage, coma and possibly death. The plasma is needed to circulate the red cells and deliver the oxygen throughout the body. So this is the basic structure of this dextrane, which will act as a plasma expander. So next one of the important one is inulin. Inulin is a polymer of the fructose monomer. It's made up of the fructose. The beauty of this is the urinary insulin clearance is considered the gold standard for the measuring of the glomerulus filtration rate, because inulin has all the properties of an ideal marker. It is freely filtered by the glomerulus, is not secreted or reabsorbed in the tubes, and it is not synthesized or metabolized by our kidney. So here by using this inulin we can say like what is the extent of your kidney is working, either it is of 90%, 89%, 29%, 15%, whatever it is. So here it will give the correct indication like your functioning of the kidney. So whether the kidney has to be transplanted or not is going to be determined by using this inulin, a carbohydrate. So next we'll see the biological role of the carbohydrates. Basic function is to provide the energy, and it will to feed the neonates milk sugars, the storage in plants starch, storage in animals glycogen, synovial fluid as a shock absorbing, anticoagulant, heparin in cornea, the plasma expander dextrin in detecting the glomerulus filtration rate, inulin, chondrocyte, tendons and cartilage. So here the carbohydrates are going to be present in and around of our body. So here some of the tests that helpful for the identification of the carbohydrates. Normally we'll use a mnemonic like MBBS, M for Molish test, Bendix test, Baffle test and Siemenov test. So this is basically useful for the detection of the fructoses, and moreover ozones also utilized to find out a specific category of the sucrose, sugar. Okay, hope this video is useful. Thank you.